move on to today's topic. So today we're talking about differentiation of T cells. All right, that's what we're talking about. So differentiation of T cells. Okay, so to give you an overview of T cells, we said earlier that both B and T cells start in the bone marrow. Okay, B and T cells, they are born in the bone marrow. Okay, so but the B cell stays back in the bone marrow to mature. All right, and the T cell will leave the bone marrow, as you can see here. T cell precursor will leave the bone marrow to the thymus. All right, so we said the bone marrow and the thymus are both primary lymphoid organs. All right, so the B cell stays back in the bone marrow, the T cell will move on to the thymus. All right, so once the T cell precursor, so that's the T cell precursor. So why is the T cell going to the thymus? Because that's where it goes to complete maturation and development. It has to go to the thymus. Otherwise, if anything disrupts this process of T cell going to the thymus, we've seen some conditions that can result from that. Number one, the George, for example, where if the thymus is not there, then the T cells cannot develop, and so the patient will come down with T cell dysfunction and be prone to all kinds of infections. Okay, so let's look at that in detail. All right, and okay, before we move on to the details of it, once it's done in the thymus, once the T cell has become fully developed, okay, that's these cells over here. Okay, so once it's fully developed, they will migrate to the lymph node. Okay, the secondary lymphoid organ. And you can think of the lymph node as the battle station where the T cells will go and get ready to encounter antigens that will be brought to the lymph node. All right. And then once they are stimulated by an antigen, okay, they will now leave and enter the peripheral blood and head to wherever they need to go to fight off the infections. Right. So, like I said earlier, there are two main types of T cell. There's the cytotoxic T cell, and then there's the CD8, all right, and then the CD4 T cells. Okay, now that we've seen that overview, let's go into more detail. So we said the T cell that leaves the bone marrow to the thymus is called the T cell precursor. Okay, so that's the one that leaves the thymus and gets to it leaves the bone marrow to the thymus. So once it gets to the thymus, all right, it's it first reaches the cortex before going into the medulla. Okay, it starts in the it arrives in the cortex of the thymus, and then from the cortex will enter the medulla. Okay, so once the T cell precursor reaches the cortex first, all right, then goes into the medulla of the thymus. All right, and the T cell that arrives in the thymus, in the cortex of the thymus, is this cell. So you can see it's double positive. They call it double positive. Okay, double positive. Why? Because it has both CD8 and CD4 markers on its surface. Okay, remember I told you that no, if you if you check the blood of measure the T cell, look at the T cells in the blood of every normal person, you're supposed to see either CD4 or CD8. But during development of the T cell, when it gets to the cortex, all right, the cortex of the thymus, that immature T cell is known as a double positive immature T cell. All right, double positive immature T cell. All right, so that's what they call it, double bot, because it has both CD4 and CD8. So this is what is in the cortex. Now, there's a process that happens here called the positive selection, which we're going to look at now. All right, so we're here in the cortex right now. Okay, so what happens in the cortex? So we're in the cortex now. So there's a process called positive selection. All right. So this is going on in the cortex. And what I would like to say before we look at the details of this positive and negative selection, All right. Just imagine that you have a pharmaceutical that is developing a drug. Okay, that is a disease out there, any disease you can think of. All right. Let's say maybe malaria or so. And they want to develop a new drug. They're trying to look for a drug that is better than what they have, or they're trying to develop something. They're going to concoct a whole series of different kinds of drugs, okay, to test those series of drugs to see if there's any of them that has any effect on that disease. Okay. So the ones that they can measure an effect, that has any measurable effect on patients with that disease, right? They will select those ones, those different type of drugs from the concoction that they've made, from the series of concoctions they've made. If there are any of those drugs in those series that have any effect that can somehow reduce the severity of the disease by any measurable effect in the patients that are suffering from that disease, they will, they will select those ones for further testing. All right. So that's what you would say positive selection. Those will positively remove the ones that have some effect on the disease, okay, that reduce the severity of the disease to any measurable effect out of the whole series of drugs they've taken, out of the whole series of drugs they are testing. So that you say that they positively removed the ones that can have an effect on the disease. Okay. Then once they've selected the ones that have some effect on the disease, they'll now go further to test those new set of drugs they've removed. Okay. So you had a whole series here of drugs, okay, for the disease. Then the ones that had some effect, okay, they now positively selected those ones, positive selection to now have a smaller series, a smaller number of drugs, okay. Then they will go ahead and test these ones, all right, to see how whether they are safe, okay, and the side effect profile and whether how harmful they are. So the drugs that have severe side effects or that cause more very harmful side effects or are very bad for the patient, they will now negatively remove those drugs. Those drugs will be removed. Okay. So the ones that will be left behind, there are many drugs that will be left behind, will be the ones that are the best, that have the safest profile for the patient. This will be the safest and probably the most effective. They may not be the most effective, but they will be the safest. Okay, they are not going to, in terms of side effect and all those, they cure the disease or they'll treat the disease, but at least, and more importantly, they are not harmful to the patient. Okay, so I'm walking through the positive and the negative selection. So coming back to, just to give you an example of what the thymus is doing, because in the thymus is actually screening the T cells that come in. Okay, those immature T cells that are coming in, is screening them to weed out the T cells that will be potentially harmful to the body. All right, so that's what is happening in the thymus. When those immature T cells come in to develop in the thymus is weeding out the T cells that are going to cause any problem in the body. All right, so that's and the process of it doing that is the positive and the negative selection. All that process is to weed out harmful T cells. And what do I mean by harmful? T cells that can lead to autoimmune disease. When you say autoimmune, okay, when we say autoimmune, okay, we're talking of immune cells that attack the body. All right. So if you think of any autoimmune disease, that the ones we've looked at previously, okay, it could be in the GI, the thyroid, in the brain, in the bones, the kidney, the heart, any or virtually almost any organ can be affected. All right. So during T cell development in the thymus is trying to screen out and remove those potentially harmful T cells that down the line, if they are not removed, they can lead to autoimmune diseases. All right. So that's the main thing that the thymus is doing. All right. So let's look at what happens once the T cell gets to the thymus. So in the cortex here, it's the T cells that self-recognize peptides are allowed to allow for incorporation in immune responses. So the double positive thymocytes expressing T cell receptors that recognize self MHC complexes receive a survival signal. So what does that mean? So like I said earlier, if you talk, I gave you the example of the company that is screening, looking at the different series of drugs to see if there's any of them that works on the disease. The same thing, the thymus is screening the T cells that are coming in the cortex and seeing, okay, these T cells that are coming, they have a T cell receptor on their surface. This is the T cell receptor that binds to antigens, the antigens on the MHC that we talked about, whether MHC1 or MHC2. All right. So the T cell receptor is the receptor on the T cell that interacts with the MHC, the antigen on the MHC. So the thymus is presenting antigens to the T cells, the immature T cell, to see if there's any of them that has a T cell that works, if it's even capable of, if the T cell receptor even works, if the T cell has a receptor that works at all, that is capable of even recognizing any antigen. All right. So if you have it, if there's a T cell that works that can recognize an antigen, that means the T cell receptor is functioning. Then that cell receives a survival signal. That T cell receives a survival signal and can go down to the medulla. So that means at least it works. It's functional. That means if it goes out into the wild and APC, an antigen presenting cell, presents an antigen to it, it can recognize it. That's the implication of that, all right? So, but for now, I'm just saying that the the double positive thymocytes or T cells that come in, their T cell receptor is being evaluated to see if it works. Okay, and the ones that can recognize self-peptide MHC complexes will receive a survival signal. So those ones will go down into the medulla, right? So now coming down to the medulla, this is in the medulla now. So this is the cells that have reached the medulla are the cells that receive the positive survival signal in the cortex of the thymus. So those are the ones that their T cell receptor is functioning. All right. So the ones that their T cell receptor were unable to bind the self peptide MHC, all right, will die off. The ones that means that T cell receptor is not functioning, and so no need to take for them to move any further. All right, so moving down to the medulla now. Okay, in the medulla, T cells that bind too strongly to self peptides will die off or be weeded out. And why is it weeding those ones out? Because these T cells will react strongly to self antigen. That's what this statement means. Okay, so T cells that react too strongly to self antigen. When we say self antigen, we're talking of antigens of your body, of your human body. They are supposed to react to foreign antigens. Okay, not attack self antigen. They're supposed to attack foreign antigens and not attack self antigens. All right. So the thymus is basically testing in the medulla. The thymus, the thymic medulla is basically testing those T cells to see if they are going to react too strongly to self antigen. And then if they do, if they bind too strongly to it, it will remove them. And why is it removing them? So that those T cells will not go out, leave the thymus, and then go on to attack normal tissues in the body. All right. So that's where thymocytes, when you say thymocytes, that's immature T cells, expressing T cell receptors with high affinity for self undergo apoptosis. All right. That's one outcome, or become regulatory T cells. So the T cells that react too strongly, okay. So if we say strong self-reactive T cells, two things will happen to them. Either they undergo apoptosis or they become regulatory T cells, right? Those are the two things that will happen to them. So either they will die off or they become regulatory T cells. All right. So what gene? There's a gene that drives this negative regulation, and that gene is called autoimmune regulator. That's AIRE. Okay. So it drives negative regulation. So if there's any problem with this gene, that means these T cells that react too strongly to self antigens will be allowed to escape from the thymus. So these cells can escape from the thymus and go into the blood and travel to different organs, and then they can lead to autoimmune conditions. So one condition that results from this area is called, I mean, playing this. So one condition that results from this deficiency of this enzyme is called autoimmune polyendocrine syndrome. Autoimmune polyendocrine due to a mutation in the AIRE gene. Okay, so negative, if that happens, negative selection does not happen. So T cells that can react very strongly to self antigens are allowed to escape the thymus and go out and can cause problems by attacking different organs. Okay, so now you have chronic, the conditions that make up this are chronic mucocutaneous candidiasis, all right, hypoparathyroidism, adrenal insufficiency, and recurrent candida infections. Okay, recurrent candida infections. So those are the you have the pneumonic here. Without air, your body will char. Okay, so the C is for the candidiasis, H for the hypothyroidism, A for the Addison's or adrenal insufficiency, and R for recurrent candida infections. Okay, so those are the conditions that result from the deficiency or mutations of the AIRE gene. All right, so if AIRE is mutated, negative selection does not happen, leaving autoreactive. That's autoreactive. When I say autoreactive, that just means T cells that are capable of attacking the body, normal tissues of the body, were allowed to escape, and then all these symptoms, all these conditions will result from that. Okay, so now moving on. So we are done with positive and negative selection, right? So we have left the thymus now. Okay, we are now down here. So this macrophage lymphocyte interaction is what happens here in the lymph node. Okay, so the normal process has gone on in the thymus. Your CD8, okay, your CD8 and CD4 positive T cells, all right, these are the cells here, have now have graduated from the academy. Okay, they are okay, they are normal, they are fully functional. They become the cytotoxic and the helper, the cytotoxic CD8 positive T cell and the CD4 helper T cell. Okay, these cells will go into the lymph node. All right. So once they get there, ready to interact with an antigen, a foreign antigen, this is where we are now. They are in the lymph node here. This is in the lymph node. Okay. That's where they will meet the antigen presenting cells. All right. In the lymph node. So we said the antigen presenting cells, we said from yesterday's discussion, we said there's the macrophage, okay, the dendritic cell, said the B cell, said, and the what's the Langerhans cells. Okay, these are the cells waiting for them that the T cell will encounter in the lymph node and get and through them, through these cells, the T cell will now encounter a foreign antigen. Okay, so on its own, the T cell cannot see a foreign invader or any antigen unless the APCs bring it to the T cell. Okay, so the T cell on its own cannot go out and see or notice a foreign antigen. It has to be brought to it by the APCs. The APCs are the eyes and you can look at the APCs as the eyes and the ears of the T cells. Okay, particularly the CD4 T cell. Why we CD4? All right, because the APCs they interact with the CD4. All right, so now this macrophage lymphocyte interaction now, and this interaction now is going to be this macrophage lymphocyte is more for the CD4 T cell. Okay, it's the one that interacts with the APCs. So the APCs are the eyes and ears of the CD4 T cell. So the CD4 T cell will not know what's happening if the APCs do not bring any information to it. All right. So that the APCs are the ones that will tell it what's going on, what type of pathogen, where it's coming from, give it all the lowdown, all the information it needs, and then its CD4 will tell the other cells what to do. Okay. So now that brings us to this. Now we're looking at the CD4 helper T cell. Okay. Now, this macrophage lymphocyte interaction, they're talking about this interaction. Okay. So let me see. So when the macrophage interacts with it, now let me see. I think I should have a diagram that's illustrates that. Okay, so look at it here. So this is your. So look at the macrophage here. This is the macrophage. Okay. So my pen is a bit okay. So this is your macrophage here. All right. Let me this one. So this is the macrophage. All right. And then it's brought an antigen. Look at the antigen right here and presented it to the CD4. This is your CD4 T cell. Okay. So the other thing I forgot, I just once the cell before they interact with an antigen, before they become activated, they are called the naive T cell. When you hear a naive T or B cell, this word naive, it just means an unactivated lymphocyte T or T cell in this case. All right. When they say naive, it means it has not encountered an antigen. It has not yet encountered an antigen. Now, after this interaction with an antigen, then it becomes activated. So these are all activated T cells. All these ones you are seeing here, this one, they are all activated T cell. But before it became, without any antigen presented to it, in the absence of an antigen presented to it, it's called a naive T cell. All right. So now this is happening in the lymph node, or you could say the spleen as well, in the secondary lymphoid organ. Okay. So the APC, whether the dendritic or the macrophage, all right, it brings the antigen to it, which you can see here, presents it to the T cell. So the T cell receptor, this is the T cell receptor, will bind to that antigen. This is the MHC. This green particle you can see as the MHC2. Okay. So the antigen is mounted on the MHC2 on the APC, and then the APC will bring it and then to bind to the T cell receptor on the naive T cell. Now, so here the cytokines are produced. Okay, like I said, cytokine is like it's a language that cells use to communicate with each other. All right. So the macrophage will release the cytokine to tell the B cell, okay, hey, or the T cell rather, hey, look, I have something to show you. I need you to take a look at it and let me know what you think. All right. So it's activating or informing the T cell that look, I have something to show you. You need to take a look at this. That's what the cytokines are for. All right. So, and it depends on the type of cytokine that this cell APC releases will also, in a sense, the type of language that the APC speaks will determine the response of the T cell. All right. So different cytokines or different languages will elicit different responses. That's what I mean. Okay. So in this case, if the APC produces interferon gamma, this interferon gamma is coming from the APC. All these ones you're seeing up here are coming from the APC. Okay. Okay. No. Sorry. I'm making a mistake here. Okay. So the cytokine that induces this subset. Okay. So this is coming from the APC. So if the APC produces interleukin 12, all right, and interferon gamma, that's from the APC to the T cell. If it produces interferon gamma or interleukin 12, it will induce this naive T cell to become a Th1 cell. Okay. If the APC produces interleukin 4, okay, we're talking. I think there's a full chapter on cytokines in first aid. We're going to come to that. It's going to be a topic we'll probably take the whole day discussing. Now, this is just a preamble. So I just want to give you some ideas of it before we get to that point. Okay. But just for you to understand that the cytokines induce the cells to do certain things, and the action of the cell depends on the source as well as the type of information it's receiving. Okay. So it behaves a certain way based on the information it receives. So different information elicits different actions. Okay. So that's the point I wanted to make. So if the naive T cell receives this interferon gamma and interleukin 12, it will induce it to become a a a Th1 cell. All right. If it receives interleukin 4, all right, it becomes a Th2. If it receives this ones, it becomes Th17. So now that we've looked at that, let's move on here. So that's those cytokines are activating the naive T cell to become a specific type of cell. So look at them up here. Okay. So if you look at this, the interferon gamma interleukin 12 we talked about coming from the APC will activate this helper T cell, naive helper T cell, because this is a naive cell here. Okay. To activate this naive cell, naive CD4 cell to become a Th1 cell. All right. So depending on the cytokine that the naive helper T cell receives, it can become either a Th1, Th2, Th17, or Treg. It has 4 possible outcomes, and they all depend on the type of cytokine that comes to it, the type of information it receives. All right. The type of information it receives. All right. So that information is what you're seeing here. The activating signals are the information that it will receive. All right. So let me clean some of this off so that we can get a better view of what we're looking at here. So if we're focusing on the first one, that's the Th1 cell. So what signals will induce the naive CD4 helper T cell? That's the interferon gamma and the interleukin 12. Okay. And these ones come from the APC. So the APC, like we said, will present the antigen. Okay. Look at it here. We'll present the antigen to the naive CD4 helper T cell. All right. And then produce, as it's presenting this antigen to it, it's also producing interferon gamma, interleukin 12. So interferon gamma and interleukin 12. Okay. Interferon gamma and interleukin 12, which will induce the cell to become a Th1. And then when it becomes a Th1, all right, this Th1 cell will go on to produce interferon gamma and interleukin 2. So we're just talking of Th1 now. So the interferon gamma and interleukin 2 will induce the cell to to induce the naive T cell to become a Th1. The interferon gamma and interleukin 12 from the APC will induce the naive T cell to become a Th1 cell. It's still a CD4 cell, and then that Th1 cell will now produce interferon gamma and interleukin 12. So the cytokines the Th1 will produce will activate a certain group of white blood cells. Okay, namely for Th1, it will activate macrophages and cytotoxic T cells. All right. So also, don't worry if you have any questions. Just hold on. Once we get to the end of this page, you can leave. You can ask me your questions. Okay. So now moving on from here. Okay. So we're done with Th1. Okay. So it's the same principle for the others. All right. So if this is your naive CD4 T cell, right? So now let's come this way. If the APC presents an antigen to it, okay, an antigen to it, and then produces interleukin 2, all right, and interleukin 4, it will induce the naive T cell to become a Th2. Now, this Th2 cell is very important in allergies and hypersensitivity reaction. Okay, hypersensitivity reaction. So Th2 is more important in hypersensitivity. Th1 is more important in infections like viral infections because or tumors or cancers because cytotoxic T cell can be activated, and certain kinds of infections as well. I said viral, another one TB, okay, viral infections, cancers, and so forth. All right. Th2 is much more relevant for allergies, hypersensitivity reaction, and parasitic infestations as well. Right. And so Th2 will produce this set of cytokines, interleukin 4, 5, 6, 10, 13, that activate eosinophils and also stimulates B cells to produce immunoglobulin E. So you can see, depending on the on the information that the naive CD4 cell receives initially, okay, that's the naive CD4 here, it will induce it to become a specific type of T cell that will now go on to activate unique sets of white blood cells. Okay, so depending on the signal it gets, it will determine its action. All right. So moving on, if we now come on next to now move on to this, the third type of CD4 cell that can be produced here, the Th17 Th17 CD4 T cell. All right. This one stimulated by transforming this TGF is called TGF is transforming growth factor beta, interleukin 1, interleukin 6. All right. Those ones will stimulate the cell to become a Th17. And this Th17 specifically, this one specifically stimulates neutrophils. So when you hear Th17, think neutrophils. So most especially bacterial reaction and bacterial infections or invasive fungal infections. All right. Invasive fungal infection, that's fungal infections that have entered the bloodstream and are spreading all over the place. So when you hear Th17, think neutrophils. And then it produces this set of cytokines, that's interleukin 17, 21, 22. All right. So that's for Th17. Next thing, let's move to the last one. All right. Yeah, I can use a different color. Okay. So the last one is the Treg. Okay, Treg. So the T CD4 helper T cell. Remember I said earlier, the Treg can be produced during negative selection in the thymus. Okay, there are cells that react too strongly to self-antigen. They can either die by apoptosis or become Treg. Another scenario where Tregs can be produced is where the naive CD4 helper T cell antigens are these cytokines are produced transforming growth factor beta and interleukin 12. I mean interleukin 2 to stimulate the CD4, the naive CD4 cell to become a T regulatory cell, okay, or a regulatory T cell. And then that regulatory T cell will produce these ones, transforming growth factor beta, interleukin 10, interleukin 35, which prevent autoimmunity and maintain tolerance. Another function of these cells is they also uh, these cytokines that you see here are also anti-inflammatory cytokines. So they help to suppress inflammation. Okay, that's anti-inflammatory. Okay, they're anti-inflammatory cytokines, so they suppress inflammation. All right. Okay, so that's for all of them. So just know that the signal the CD4, the naive CD4 T cell receives will determine its behavior or its actions. All right. So that now explains. So that diagram we've looked at up there, okay, explains what's going on here. Where once the naive CD4 cell, so this interaction is talking about this first. Let me clean this one off. So that interaction is talking about this one. All right. When the macrophage, which is an APC, interacts with the naive CD4 cell, all right, that will induce the cell to become, because the macrophage is also an APC, right? So when it interacts with the naive CD4 cell, it will induce the naive CD4 cell to become a Th1 cell, all right? And that Th1 cell secretes interferon gamma, which you can see here, all right? Secretes interferon gamma and enhances the ability of the macrophage to kill microbes they have ingested. All right. So if we go back to this diagram over here, okay, what I said is this one, if it produces this interferon gamma, so this interleukin, that's interferon gamma, okay, or interleukin 12. Then this one as well will go on when it becomes a Th1 cell. Okay, this Th1 cell will now secrete interferon gamma that will go on to activate this cell, the macrophage here in this case. And you can see the microbe it has ingested. All right. The macrophage, it brought it ingested it, took bits of it, and presented it to the naive T cell, secreted interferon gamma and interleukin 12, which stimulated the naive T cell to become a Th1. Okay, while they are still interacting with themselves, then the Th1 cell now says, okay, produces this interferon gamma to stimulate this macrophage to activate it and destroy this pathogen that it has swallowed up. All right. And then the macrophage will go back to where it came from, where it got that pathogen, and go and begin attacking the pathogen, the source, wherever it picked up that pathogen from, to go back there and start attacking more of the pathogens where they came from. All right. So that's what this interaction is talking about. So this function is also enhanced by interaction of T cell CD40 ligand with CD40 on the macrophage. So what you're beginning to see is there's a lot of interaction between these white blood cells. All right, a lot of interaction between them. So besides, and we'll look at that in the next page once we get there. So besides the MHC2 and the T cell receptor, okay, so you have the MHC2 on the APC, binding to T cell receptor. Okay, that's one interaction. Now we're seeing another interaction for between both the macrophage and the T cell. That's CD40 ligand. When you hear CD40, it's just a molecule. It's a molecule on the surface of the T cell. It binds to CD40 on the macrophage. Okay, and we'll come to see there are other interactions like this that have to happen at the same time. Otherwise, the macrophage will not get the activation it needs from the T cell, or the T cell will not be induced to differentiate into a Th1 cell. We'll come to see that all these interactions are essential, and they are disease processes that result if any of these interactions is disrupted. All right. So let's move on. And then the next line here says the macrophage also activates. So this is from the it also activates the lymphocyte via antigen presentation. So this involves MHC2 on the macrophage. All right. So we're done with now that we're done with that statement, we're done with this line. Okay, so we're done with CD4 cells. Now we're moving on to cytotoxic T cells. All right. So for cytotoxic T cells, we said they kill virus-infected cells, neoplastic cells, and donor graft cells by inducing apoptosis. Okay, using perforins and granzymes. Okay, so they release cytotoxic granules containing preformed proteins. Okay, that's the perforin and the granzymes. All right, so the cytotoxic CD8 cells have CD8 on their surface, which binds to MHC1 on virus-infected cells. So this is not there's nothing really new here. We've already looked at this. We talked about CD8 in the previous section. Just like the natural killer cells in terms of using perforins and granzymes. Okay, but they need the MHC1 to recognize virally infected cells. The NK cell does not need MHC1, okay, to recognize bad cells. Okay, so we're done with T cells. That's CD4 and CD8 T cells. Now we're moving on to regulatory T cells. Okay, so they help to. So regulatory T cells, like we said. Sorry, can you wait till when we get to the?
Okay.
Sorry, just wait when we get to the end of the page. Mute your mics. Please mute your mic. Okay. So we said regulatory T cells, they help to maintain specific immune tolerance. So when we say immune tolerance, this time immune tolerance means the body's own immune cells. Look at your head. You are talking about the T cells will not attack the body. Okay, the normal cells of the body. That's what is called immune tolerance, to not attack your own tissues. Okay, to live so that the immune system is able to live in harmony with the cells of the body and not attack it or see it as foreign and attack them. So that's the function of the regulatory T cells. All right, they will suppress the CD4 and CD8 T cells so that they don't go and attack, because if they do, that will lead to chronic autoimmune disorders that will be debilitating and can lead to organ damage and eventually death. Okay, so what are they identifying? What's the identifying marker of this T regulatory or regulatory T cells? They have the CD3, CD4, CD25, and FOXP3. So this is very important. All right. FOXP3, we'll come to look at it in detail shortly. All right. So the activated T cell produces anti-inflammatory cytokines. So like we talked about. So we talked about it here. All right. The regulatory T cell produces your anti-inflammatory cytokines. Very important that you know this, because you get tested on this. So transforming growth factor beta interleukin 10, they are examples of anti-inflammatory because they will suppress the action of the other immune cells like CD4 and CD8 T cells. All right. Now, this condition we're looking at here, IPEX. Okay, that's immune dysregulation. All right. Polyendocrinopathy, that's IPEX is an abbreviation that means I for immune dysregulation, P for polyendocrinopathy, E for enteropathy, okay, and it's X-linked, meaning if it's X-linked recessive, then it occurs most likely in males, okay. So IPEX, and it's due to a disorder in FOXP3. So we already said here that FOXP3 is present in regulatory T cells. So if you have a disorder of FOXP3, that means your regulatory T cells will go down. All right. If they are down or absent, that means no one to suppress the CD4 and CD8 T cells. All right. So they just keep going, 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 going. It's like they are just constantly activated and they will attack different tissues and organs, and that will lead to autoimmune conditions. Okay. That's why I say autoimmunity here. And it's characterized, like we said earlier, enteropathy, that's disorders of the GI, endocrinopathy, nail dystrophy, dermatitis, all right, and autoimmune dermatologic conditions. So very important you know this. So enteropathy probably from GI, I mean from diarrhea, probably chronic diarrhea, such, and maybe to lead to malabsorption. So they cannot, so these people will be malnourished. Okay, endocrinopathies involve virtually any endocrine organ. Okay, the nails are look disordered and malformed, okay, or disfigured nails, dermatitis, like for example eczema, because the T cells are attacking the skin, and then other autoimmune dermatologic conditions, and it's associated with diabetes in the male infant. So this typically occurs in new young children. Okay, this FOXP3 problem occurs in young children. All right, so you see a young child with diabetes, and then you see eczema, disfigured nails, malabsorption with probably GI, diarrhea, and then some endocrine disorders. So you should start thinking of FOXP3 deficiency, and would leading to, so if FOXP3 is absent, that will lead to absence of T regulatory cells or regulatory T cells, and therefore the activity of CD4 and CD8 cells will go up. So no one to suppress them, and they will attack other organs, normal tissue and organs, leading to autoimmune disorders. Okay, so before I close, leave this page, I want to go back and mention about this interleukin 12. All right. So why am I mentioning it? Because it's particularly important. If you're going to see questions on that that will say, okay, this. Let's go back to this diagram here. Okay, you're going to get a question that will say this. Let's say this is the macrophage, an APC. Okay, and this is your naive T cell, right? So the macrophage has brought the antigen to the naive T cell and has produced interleukin 12. Okay, it has produced interleukin 12. The macrophage has produced interleukin 12 to activate the naive T cell so that it can respond to the antigen that the macrophage is presenting it to. But in some conditions, the receptor, all right, maybe I can use a different color, all right, the receptor on the interleukin 12 receptor, this is the interleukin 12 receptor on the T cell, the naive T cell, is dysfunctional. All right. The interleukin 12 receptor on the naive T cell is not working properly, meaning that it's just like you have someone that is deaf. Okay. No matter how loud you shout, the person will never hear you. Same thing. The naive T cell interleukin 12 is like the ears of the naive T cell to hear the information, that's the interleukin 12 cytokine, all right, that the macrophage is producing to stimulate the naive T cell, all right. But because the interleukin 12 receptor on the naive T cell that's supposed to bind to the interleukin 12 from the APC or the macrophage is dysfunctional, that means the naive T cell will never respond to this interleukin 12. So it's almost it binds to the antigen, but nothing happens. All right, because it's not responding to the interleukin 12. Why? Because the interleukin 12 receptor on the T cell is bad. So that means this T cell will never get activated. All right, will never get activated to become a Th1 cell. It will never get activated to become a Th1 cell. All right, so it will never get activated to become a Th1 cell, and so that means if it doesn't get it to become a Th1 cell, that means it will never produce the interferon gamma here needed to activate this macrophage in order to destroy this pathogen that it has swallowed up. So the question will say, all right, so okay, so the question will go that, okay, so like we said here, it will, if it doesn't, if this interleukin 12 is unable to interact, unable to activate this naive cell, it will never become a Th1 cell. Okay, and then interferon gamma is never produced, and therefore the Th1 cell can never activate the macrophages or the cytotoxic T cell. So it will always be stuck as a naive T cell because it cannot bind to the interleukin 12 coming from the macrophage that is presenting antigens to it. So the question scenario will be that, okay, you have a person that is having recurrent mycobacterial infections. Okay, mycobacterial infections. And then they did a testing and they realized that the interferon gamma is low, okay, or absent in that patient. And then they'll ask you, what's the problem? So if it's low or absent, it means that there is no Th1 is the most important source in the body for producing this cytokine. It's the most important source in the body for producing this cytokine. So if you don't have a Th1, you won't have interferon gamma. All right. So, and that means the naive T cell didn't get activated to become a Th1. All right. And then the most important source to activate the naive T cell to become Th1 is interleukin 12. So that means something is wrong with the naive T cell. All right. So that's the question for interleukin 12 receptor deficiency or disorder. All right. I'm hammering on that now because you're going to encounter it once we move on, and you're going to see questions on it. So it's something you should remember. Well, there are other ones, but at least for now, it's just a preamble to what's coming ahead. All right. But since they've brought it up here, so that because once you see it in the future, as you move on, you may not understand why are they talking about interleukin 12 receptor deficiency. How does that lead to immunodeficiency, particularly with TB? Okay, because the macrophages are the most important cell. Okay, we said here the macrophages are the most important cell that handle intracellular pathogens, particularly TB. And so when they encounter TB, they go to the naive CD4 cell and try to stimulate it, producing interleukin 12 to stimulate that naive CD4 cell to become a Th1. They need that naive CD4 to become a Th1 so that it can go stimulate them back, the macrophage, and activate it to go back and deal with the TB pathogen. And the only way, and the thing that activates the macrophage is this interferon gamma that you're seeing here. All right, is the interferon gamma that you're seeing here. So the macrophages need this to become activated to go back and deal with the mycobacterium pathogen. So this is a condition that happens in interleukin 12 receptor deficiency. All right.